Start the proceedings for today.
Now the meeting is being recorded. Thank you, Pankaj. And I first of all thank Dr. Harish Murjani, my dear friend and classmate, and right now more than classmate, he's an expert, and he's on our board to discuss the current scenario of the vaccines. There has been a lot of misinformation campaign going on about the vaccine for the last few weeks, and a lot of groups have got activated. Previously, it was a challenge to develop a vaccine. Now, what I find is that the vaccine is there in the market, and some countries have already started injecting the vaccine in the frontline workers. But just at the onset of this vaccine campaign, another group of people have got activated who feel that the vaccine is likely to be dangerous and should not be given. And WHO is also working on a campaign soon to be released so that the public and the other doctors can be made aware that the vaccine is likely to be helpful to them, and they should not fear taking a vaccine. And there are another issue is that now it appears a problem of plenty. There are 4 or 5 vaccines in the market which are there already or are likely to be available on board soon. And doctors will be in a dilemma which vaccine to choose and which vaccine is likely to be more effective and which is more likely to be more safe. And to discuss all these issues, we have with us Dr. Harish Murjani. And Dr. Harish Murjani is an infectious disease expert. He is an MD and he is a specialist at the Hudson Infectious Disease Associates, New York. Dr. Murjani also serves as a clinical assistant professor of medicine at New York Medical College in Valhalla, New York. He also practices at Philips Memorial Hospital. And Dr. Murjani received his Bachelor of Medicine and Bachelor of Surgery degree from Moderna Medical College, University of Delhi. He has a fellowship of infectious diseases at State University of New York at Stony Brook in Stony Brook, New York. In addition, Dr. Murjani is an infectious disease consultant for the New York State Department of Corrections, Westchester County Jail for the past 2 decades. He has treated more than 1,400 patients of COVID in the last 8 to 9 months, and he has been instrumental in bringing down the mortality from 8% to 1.5%, or even less than that in New York City. And you will all recollect when we were not knowing anything about the pathophysiology of the disease. It was the New York county jail, which started doing the autopsies, and by those autopsies, we came to know that the disease is causing thromboembolism and the thrombotic phenomena in the pulmonary vessels. And from where we started learning all this, and Dr. Murjani was instrumental in delineating the pathophysiology. And we are very lucky to have him on board to talk about vaccines, the current status of various vaccines, the various scientific data of various vaccines, and which vaccine to choose. Let's listen from Dr. Murjani. Over to Dr. Murjani. Thank you, sir. You're welcome.
Hey, well, thank you, Satish. Thanks, Pankaj. Thanks, everybody, for giving me this opportunity. So my job is to actually share the information, and most of these are my opinions. Of course, they're based in science, and I'll present the data. And I think it's a very controversial issue right now, and understandably so. It's a new disease. The disease is evolving very fast, as Satish mentioned. Some of the concepts that we understood over the last 5 or 6 months have stood us in good stead. Our current hospitalization case fatality rate is down to 1%, and the overall population case fatality rate in the sphere of influence that we are in New York, is down to 0.1%. So we have achieved those goals that we set out to do. But we are still seeing 270,000 new cases every day. We are still seeing 2,900 deaths every day in the US, all over. New York, the number of deaths is much less. I understand that. And so this vaccine is supposed to bring down the amount of infection rate, for one, also bring down the spreaders and the amount of virus that is being spread, so that we can handle it much better. And until such time, and I'll also share with you a couple of new concepts of how we are going to prevent patients from progressing. We heard about a case recently, just now, a few minutes ago, about how the patient is having all the complications of COVID, how we could have prevented that early on. And I think in the next 6 months, you're going to see the face of this disease completely turn on its head. It's going to be very manageable. It's going to be mild. It's going to be taken care of. And before we do anything, I think I just want to reiterate, this virus, the COVID-19 virus, is unlike we have seen ever before, as was mentioned here, just a few moments ago. In my personal opinion, this is not a naturally occurring virus. How this virus came about with a combination of coronavirus genes and retrovirus genes, and to be able to really throw into mayhem your inflammatory and coagulation pathways is unheard of. This kind of evolution doesn't occur in nature. You see evolution occurs in a very gradual fashion. This wholesale genetic code mutation that has occurred with a 30 codon virus that we cannot even compare to anything else ever before, and which is being manifested in the pathophysiology, is something we have to understand. And I think that's key. Once we understand that and we get a grasp of that, that this is not a naturally occurring virus, this is completely different from what we've seen, then it forces us to be on our clinical best. Because if you cannot stage the patient on that 21-day cycle, from day zero of where the patient acquires the infection to day 21, when all the complications start to set in, post-COVID complications, then you will not be able to manage this disease. And the disease goes through various phases. We learned that last time. And it's come out true. Everybody else is now recognizing it. We have evolved from that time, about a month and a half, 2 months ago, when we did the last update, where we now are adjusting doses and duration of steroids and doses and duration of anticoagulation based on clinical markers and based on lab markers. So that is the new evolution. We are also now realizing that neutralizing antibodies, and I'll show you some data on that, used early on in the disease can actually prevent progression of disease by neutralizing the virus. So that's the other thing. And then we are now into what I alluded to last time, into the era of preventing the cytokine storm to begin with. So forget about steroids. Steroids we know are a double-edged sword. So we are trying to reduce as much steroid use as possible. And neutralizing antibody is one way, and then JAK inhibition, which I alluded to last time, is another way. So I will talk briefly about that. Most of this, what I'll talk about this time, is going to be on the vaccines, and such an important issue. So, it's magical to see that Operation Warp Speed, which lit a fire under this whole vaccine initiative. In 10 months, we now have an effective vaccine, and a vaccine that is blowing away expectations of even the most people who thought the vaccine was not possible. And it is due to one technology, and that's mRNA. So I'm going to start. Satish, can I share the screen? Let me see if I can share the screen with you guys. Here. Share. Okay. Oh, why am I not being able to? All right. Everybody can see that, right, Pankaj?
Yeah, it's fine, sir. It's fine, Dr. Harish.
Good. Okay. So this is, of course, the time course, which I alluded to. And this is everybody who's dealing with COVID-19 should be very familiar with, which is the viral phase, which is the host inflammatory response phase, what happens in the pulmonary phase, what happens in the inflammatory stage, and what happens is the clinical presentation, what are the lab markers, where can you use what intervention. So remdesivir, which the Solidarity trial was trying to trash. I mean, that Solidarity trial, again, Hindi mein kya bolte hain, naach na aaye, aangan teda. If you don't know how to use the drug and you don't know how to manage the disease, don't blame the drug. The drug is perfectly fine when used properly. And we have shown that with the case fatality rate down to now 0.1%. And so dexamethasone clearly works, but not in the way the British initial study, the Survive study, whatever the study was, I've forgotten, the recovery study showed that the dexamethasone, 10 days of dexamethasone actually lowers death rate, but it doesn't lower it down to 1%. It lowers it down to like 18% or 17%. So we went further. We looked at C-reactive proteins and the C-reactive protein correlation with inflammation and how much decadron to use and how long to use it for. And we've been very successful with that. Oxygen, we've now realized that gentle ventilation, as was pointed out before, is the way to go. So a lot of this stuff is going on, and it has to be used appropriately at the right time. If you do the cookbook medicine stuff, what is being taught in the Western countries recently, lab-based medicine, cookbook medicine, throw everything at everybody, it's not going to work. You have to do the right intervention at the right time. And I think that's the key to bringing the case fatality rate down. So I'm not going to spend too much time on it, but here is what I was talking about. We are looking at the monoclonal antibodies. This is the Eli Lilly model, which is a single monoclonal antibody against the spike protein, but it's not the entire spike protein antibody. It is just the antibody that binds to the binding domain, the receptor binding domain, which is the part 1 of the spike protein. Part 2 of the spike protein has to do with the fusion. And I think that's very important to understand that, because that's what leads to antibody dependent enhancement sometimes of the virus, and we have to be very careful about that. But here's clearly the study that's showing that using it in the first 7 days, so you assume that the infection occurred 4 days before the test, and within 3 days of that, if you infuse it, you're going to reduce the severity of disease and reduce hospitalizations. So you take the pressure off of the hospitals, and patients do much better. This is the one that, this is not the one that Donald Trump got. He got the Regeneron one, which I'll show you the data on. But so this is a single monoclonal antibody against the RBD. And then, so the FDA actually granted a authorization for this to use. It is not authorized for use in hospitalized patients. It has to be used before the patient gets to the hospital. They should not be requiring extra oxygen. Their oxygen level should be above 94% for one, unless they already have low oxygen to begin with. And so, and they have to have either a BMI more than 35, CKD, immunosuppressive disease, or on an immunosuppressive treatment, 65 years of age. So they have to be at risk for progression. You don't want to give it to somebody who's going to recover naturally anyway. There are other interventions for that subgroup of patients. So patients 12 to 17 years of age with more than one of the following, patients more than 55, very clear, concise, guidance. And we've been using it now. We've used this Eli Lilly monoclonal antibody, at least under my care, in more than 25 patients. And I'm happy to report that all 25 have been cured without any other intervention, period. So I had a couple of doctors recently last week who got it, and one of them texted me this morning that he's back to normal. He's going on his run this morning around the lake reservoir. So, great news from that. Now, Regeneron is a double monoclonal antibody, both against the part 1 of the spike protein and also part 2. So it not only inhibits the binding of the virus to the receptor binding domain, but also prevents the fusion channel where the virus gets into the macrophage cell. So it's important, this is the one that Donald Trump got. This is the one that Rudy Giuliani got. So clearly the drug it works. It reduces hospitalizations by 72%. It reduces severity of disease in almost 95% of patients. So again, trying to take the pressure off of the hospitals. We know that mortality of this disease is much higher in hospitals that are short-staffed or overwhelmed with COVID-19. So if the ICU goes from 10 beds in one small community hospital to 50 beds, the hospital cannot manage, and you're going to see bad complications. You're going to see, fall off in quality of care. So this is designed, these two monoclonal antibodies are designed. So you will see a lot more about this in the coming months. And this is the basis for vaccination. And that's why I present this up ahead. So what about vaccine development? I'm not going to spend too much time on it. Most of us know this, that usually it takes 2 to 10 years for a vaccine to go from where it's first thought out in the preclinical phase to, when it first comes as approved, at least in the United States. And you can see for the SARS-CoV-2 vaccine candidates in development, there's 87 in preclinical 1, 38 in phase 1, 16 in phase 2, 12 in phase 3. And approval, I say 0, but I should say 1 because we got approval on Friday. This slide deck is 1 week old. I created this a week ago. So we now have the first approved vaccine, and this is limited to, this is from the New York Times and New England Journal of Medicine. This is limited to the US, not to the rest of the world or any part of the rest of the world. So vaccine candidates in development for SARS-CoV-2, you talk about whatever platform you want to talk about. We know about the adenovirus vector. We know about the mRNA, which I will focus on today. And we also know about, the other forms of adjuvant. We will talk about the Sinopharm vaccine. I'll show a little bit of data on that. The various forms of adenovector delivery, and also then, of course, the M adjuvant, not the whole attenuated virus, but the spike protein. So those are the ones that I will talk about today, because the spike protein one, of course, is the one that everybody's focusing on. So this is the Oxford AstraZeneca study design. Again, I was a little annoyed about this when it was first announced. It was announced in the BBC. It was not announced through the scientific channels. And this was announced with a lot of hoopla that Oxford had come up with this fantastic chimpanzee adenovirus vector vaccine. They took the spike protein and they would include it into this adenovirus vector, inject it into the humans, as a single dose, and they expected that they were going to get a fantastic spike protein antibody response, and that would prevent, the disease from progressing.
Dr. Sanath, kindly mute yourself. I think you are unmuted.
Thank you.
Dr. Sanath, kindly mute because I'm not a host today, so I can't mute. Dr. Sanath, kindly mute yourself. Thank you.
Thanks. All right, so this is what was rolled out, and it was expected that this would do well. So of course, the side effect and local reactions of this vaccine were pretty impressive. As you can see, there's nothing here, nothing red. So it was all and nothing brown. So all you were seeing is these mustard yellow and the pinks, which mean usually mild reactions, mild local reactions. These were the systemic reactions. You started to see some fever, some fatigue, some malaise, but really nothing until the 2 cases of transverse myelitis surfaced 3 to 4 months later. And we know that about the adenovirus vector. This is nothing new. This is expected. You expected to see it in about 1 to 5,000, 1 to 10,000 cases. And when you're talking about billions being infected, you will see some adenovirus. What was surprising was how the neutralizing antibody response fell off. So, local systemic reactions more common. Tylenol, of course, and with mRNA vaccines, Tylenol may not work as much, and I'll tell you why. No serious adverse events related to the Oxford vaccine. Few adverse events following the second dose and prime boost subset, but the subset was very small. So this was the neutralizing antibody response at day 56. What was surprising at day 90, the response fell off. And then it was discovered that the response was falling off because they were using the same chimpanzee adenovirus vector. So after the first shot, the volunteers who got this vaccine developed an antibody to that chimpanzee adenovirus. So the second shot didn't work as well. So it didn't boost the neutralizing antibody as much and for a long period of time as was expected. So what AstraZeneca and Oxford did was something underhanded. They changed the dosing schedule in the middle of the trial. They realized that they were not going to get to 70%, 80% protection. Now, once that data gets released, they are now saying officially that it was a mistake. They gave a half dose. First, the prime boost vaccine, the prime shot was half dose, and that was a mistake. It was actually a error. There was an error in dosing. That's nonsense. It was not an error in dosing. This is not how things happen in the vaccine trial worlds. This was intentionally, they did that in 2,000 patients. And clearly they saw that with less antibody levels against the chimpanzee adenovirus vector, that you got a much better neutralizing antibody response to the second shot, which is of course the most important one. And I think that's alluded to in this, in this whole, I don't know, I thought I put it in here, but it was clear that in people who had a prior adenovirus antibody already existing, a strong correlation between neutralizing antibody responses and antibody levels measured by ELISA in people who already had a prior adenovirus antibody. So it was very clear that this was, AstraZeneca was manipulating this data. And now they've got egg on their face. They're now trying to somehow salvage this by combining with the Russian Sputnik 5 vaccine. So the Russians were very smart, and I'll show you that data. They realized that this was a wrong strategy to begin with, that this was not going to work, especially in a setting where you're looking at other vaccines showing 90-95% responses. So this is the Ad5 vector, the CanSino vaccine. This is a little bit different, but I'll talk about the Sputnik 5 in a second. The Ad5 vector vaccine against SARS coronavirus, CanSino, what they did was they took the whole virus, the whole COVID-19 virus, and they attenuated it. And then they used Ad5 vector, the adenovirus 5 vector, to deliver that attenuated virus to the immune system. So the immune system could generate both antibody response and a cytotoxic T cell response. So you could see the baseline characteristics, adverse reactions, again, very, very favorable. Not much. Now we know that live attenuated or killed vaccines delivered by Ad5 vector have a very variable response because it depends on how well you have attenuated the virus, how well you have quality controlled the Ad5 vector, and how well you have controlled the administration and the selection of patients. So we know that. We know that this is an old-fashioned model. It's an old-fashioned vaccine delivery model, and we have now science has advanced way beyond this Ad5 vectored, live attenuated or killed virus vaccine. And we know that these, the best you could expect from this is probably going to be about 75 to 80% response at 6 months, 9 months, 1 year down the road. And it was very clear in this also that Ad5 vector antibody response was very healthy at with 2 shots, day 14, day 1 and day 28. But again, you can see here at the bottom, participants with low pre-existing Ad5 antibodies had approximately 2x higher binding antibody and neutralizing antibody levels. So, if you're already immune to the Ad5 vector to begin with, you're not going to have a good response because it will neutralize the vector itself, so it won't deliver the antigen, or in this case, the virus, to the immune system to generate an antibody response. So it's very clear that this adenovirus vectored methodology is sort of become old-fashioned. And so this is the variables associated with seroconversion. You can see in people, who have older age, people who have male sex, people with pre-existing Ad5 antibodies more than 200, you can see the response rate was, much, much less, 53% less. So, it's got its own limitations. And can you use this if you have nothing else available? Absolutely. Should you use this at a time when something else which is much better, safer, and easier has come out? Absolutely not. So again, to me, this CanSino vaccine is an old-fashioned one. This is the Sputnik 5 results. And J&J, Johnson & Johnson, is using a similar sort of approach here in the United States. So I'm going to present the Sputnik 5 because this has already been released. The J&J data has not been released, so I don't want to present something that I'm not supposed to. But take it from me that this is the exact way that J&J is proceeding with a slightly different virus, and their responses, because they have fine-tuned their delivery profile, their adenovirus vector delivery system, they've fine-tuned it much better than the Russians have. So they are getting a much better response. And we should see that in the next 3 to 4 weeks. They are supposed to release that data. So this is adenovirus 26 and adenovirus 5 vectored primary boost vaccine. You give the primary immunization, and then you give the secondary one, the booster one, with a different adenovirus. And what you can see here is most common systemic local reactions, injection site reactions, hyperthermia, headache, the usual stuff that you see with any vaccine, including pneumococcal vaccines. Lab values were mild and transient, no serious adverse events reported. In general, adverse events mirrored known profiles from other vaccines that we see with the other recombinant viral vectors in most phase 2 studies. And this is, of course, the neutralizing antibody response. You can see that, once you give a booster, first the Ad26 and then the Ad5, you get a much better neutralizing antibody response. And both the regional binding domain and also against the fusion channel. So again, they were using the entire spike protein. So they were getting antibodies against both those domains of the spike protein. So J&J is going to look the same. It's going to look at 95, 94, 90%, somewhere in the 90 to 95% response rate. How long that will last, we don't know. But let me go back. The level of response here, the neutralizing antibody response here, is very, very high. And it's expected that with the decline at a rate that we have seen with some of the other vaccines, this will probably be good enough for 6 to 9 months, probably around 12 months. But that remains to be seen. So that's important to know. The J&J vaccine actually gets a much higher RBD titer and also the fusion channel antibody titer. So that will probably be good enough for a year, year and a half, but remains to be seen. This is all speculation and on my part right now, based on just the levels of decline we have seen with other vaccines. Now, this is a completely different product. And I want to present this because this is now recruiting for phase 3 in the United States. And this is probably the closest to what you're going to see in terms of safety, efficacy to the Moderna and the Pfizer vaccines that we now have available. Pfizer is available. Moderna is expected to be available in the next couple of weeks. This is Novavax. It's the nanoparticle vaccine. What they have done is they've taken their own M protein adjuvant. This adjuvant is unlike anything else we've ever seen before. And it's a proprietary protein, which nobody knows much about. I mean, there is some structure published online. I'm not sure it's accurate, but having said that, the matrix M1 adjuvant protein is so good at taking the spike protein, so the entire spike protein, and presenting it to the immune system in such a way that it generates both the neutralizing antibody response to both the RBD and to the fusion protein, but also generates cytotoxic T cell responses that at least in phase 1 and 2 look absolutely spectacular. So, here you are, no mRNA, no adenovirus vector, nothing. All you have is the spike protein, the entire spike protein, not just the 2 surface antigens, the entire spike protein. And you have an adjuvant that is absolutely spectacular in the way it presents the antigen to the immune system and generates both neutralizing antibody response and the cytotoxic T cell response. So this is something that I'm very, very fascinated by. And I think this is probably going to be one of the 2 vaccines that in the world which are going to be the most effective. And this is probably going to be the most acceptable, given the fact that you're not messing around with inserting an mRNA into somebody's genetic code. And you can see baseline characteristics. I'm not going to go into details of it, but here are the local and systemic adverse events. Again, the adverse event profile of all these vaccines is spectacularly good. I should not use adjectives, but this is great. I mean, I cannot even believe that we have a vaccine so fast against this newly emerging disease, and that is so safe. I mean, this is amazing. You hardly see any reds around here, and none of these people had any major lasting severe, whatever severe reactions there were absolutely taken care of, and patients have recovered. So that's the important part. What is also important is that they had 5 strategies. So there was a placebo. This is a double-blind, placebo-controlled, multi-pronged, multi-arm study looking at various doses. And what is clear is that 2 doses of the 5 microgram spike protein along with the matrix M1 adjuvant results in 63,000 neutralizing antibody IgG response. That is spectacular. That is just like what we are seeing with the Pfizer vaccine. And then, of course, they also showed, at least in one paper in Nature, they showed that their cytotoxic T cell responses initially, early on in week 5 and week 6 were absolutely fantastic. So, but one has to remember, these guys are they just finished phase 2. They're recruiting for phase 3. Unless something untoward happens, this vaccine should be available to us in June. But remember, many phase 3 vaccine studies failed because the dose chosen did not offer the best balance of safety and efficacy. So once you try it in 30,000, 40,000, 50, 60,000 people, you sometimes, it's not sometimes, a lot of times will see a completely different response than what you have seen with 100 patients or 200 patients. And then, of course, immune function declines with age, and this decline is likely partially responsible for the greater risk of severe COVID-19 in older adults and may lead to poor vaccine outcomes. That is why the FDA is mandating that you study vaccines in patients who are more than 65 years of age and who have other comorbidities, so you understand that the neutralizing antibody response and the cytotoxic T cell response is durable across the various groups. And that's the rationale for that. So now switching to the mRNA vaccines, the hot topic of the year so far, the Moderna one, which is going to the FDA, going for FDA review next week, and should be available in less than 2 weeks from now, at least in the United States, and I think the rest of the world as well. A lot of the European countries and England have actually pre-bought this vaccine from Moderna. Now, Moderna is a little bit different than Pfizer. So Moderna takes this mRNA. Their mRNA platform is different from the Pfizer mRNA platform. It's not as sophisticated, so it can't handle a large lipid nanoparticle, which is encapsulated with the mRNA. And so instead of having an entire spike protein mRNA in that lipid nanoparticle, Moderna was forced to use the 2 specific antigens, one for the RBD and one for the fusion channel. So it's not the entire spike protein. And that's the key difference. And that is why this vaccine, being a little smaller with a smaller imprint, can actually be stored at higher temperatures. So it doesn't have to go to minus 94 or minus 90, whatever that Pfizer one has to go to be preserved. The mRNA here is much smaller. The platform is much smaller. It is mRNA 1273. And you can see that it also requires a booster, one at day 1 and one at day 28. It's 25 micrograms. And I'll show you the data with 25 micrograms, 100 micrograms, and 250 micrograms. And here you are. This is the preliminary safety report. Again, looking pretty spectacular. You're looking at severe reactions in very few patients, and all patients in this Moderna trial in the first 3 months actually recovered from all their adverse events that they had. This is the binding antibody to the spike protein 2, which is the fusion channel, and then receptor binding domain, which is the spike protein 1, subunit 1. So subunit 1 and subunit 2. And you can see a very healthy neutralizing antibody titer. I mean, clearly, in the 100,000 range, easily in the 100,000 range. So you're seeing this with 25 micrograms, with 100 micrograms, 250. So the dose moving forward is going to be 25 micrograms. And then here you are, the cytotoxic T cell responses. So you do that by doing a pseudoviron neutralization assay or a plaque reduction neutralization assay. And you can see very healthy cytotoxic T cell responses, in the range of 500 to 1,000. And that is actually become the new norm until you see the Pfizer data, which actually blows this away. So 25 micrograms is going to be the dose day 1 and day 28, 2 doses going forward. The data has been submitted to the FDA. They're looking at 94% efficacy across the board with 30,000 volunteers. And I'll show you in a minute the spectrum of the enrollment of the various volunteers and what was their underlying profile. But here's the Pfizer one. So the Pfizer one, takes the 2 vaccine antigens. It actually takes the entire spike protein. And the 2 spike proteins that are important, the subunit 1 and the subunit 2, both for the receptor binding domain and for the fusion channel. The mRNA platform is so sophisticated and so good that it can take the whole spike protein. So instead of splitting it up into different antigens, they are able to encode this entire spike protein antigen into the lipid nanoparticle core and put it into the 30 microgram vaccine. But unfortunately, because the mRNA is bigger, and it's a bigger platform, it has to be stored at minus 70 degrees centigrade, which is, I believe, minus 94 degrees Fahrenheit. So Pfizer had this huge hurdle to get over. And it's quite amazing how they figured it out, but they did. And they were able to figure out how to store it at that temperature and transport it at that temperature. I have posted that for my Maulana Azad group, their entire support and the way they transported, they manufactured this vaccine and the way they transported it to preserve this vaccine at minus 94. And you can see here the coronavirus 2 responses in phase 1 and how spectacular these responses are with neutralizing antibody titers. These are just sky high numbers that you saw with the 30 microgram, and they also did it with 20 micrograms, but the dose going forward is going to be the 30 micrograms. And you can see that and then this is something that actually will just blow you away. If you understand, and most microbiologists will understand this, the CD4 plus T cells, which is pre and post vaccination, the spectacular rise in these inflammatory markers, which correlate with the production of cytotoxic T cell responses, and then how these cytotoxic T cell responses then control this virus and lead to lasting immunity is quite amazing. So this is the vaccine, the Pfizer vaccine, which has shown us spectacular cytotoxic T cell responses with plaque reducing assays, which are 5 times that of any other vaccine that we have, that Moderna has shown, or even the J&J one is going to show. So very exciting to see this Pfizer vaccine. Of course, the hurdle is minus 94 storage, and then you have to use it within 6 hours of opening the vial. Each vial is 5 shots, and you have to reconstitute. So you have to have trained people who know how to give the vaccine, reconstitute the vaccine, and then administer it. The side effect profile is the same as that of any other of the other vaccines I've shown you, nothing different. There's been no major issues. What did happen was, of course, you had the National Health Service the British National Health Service, which had trashed the mRNA platform in March. And officially they had come out and said, no, we are going to go with the adenovirus platform. And we think that's very exciting. And they had said the mRNA platform is unstudied. We don't know whether it's going to work. Of course, they realized that the Pfizer mRNA platform and the Moderna platform was fairly good. So they pre-bought the vaccine and they started giving out the vaccine a week ago, as we saw. And a lot of people got it. And then, of course, this guy from the National Health Service, he's some official with the National Health Service, comes out and says that 2 participants got severe anaphylaxis reactions or allergic. He didn't say anaphylaxis. He said allergic reactions. And I'm looking at this report and I'm saying to myself, man, who the hell are you trying to fool here? You can't fool me. You can't fool doctors. All you're doing is, trying to raise the hype against this Pfizer vaccine worldwide by putting out this report that 2 people have had an allergic reaction and all people with all allergies should be very careful about taking the vaccine. What a nutcase. The first thing the National Health Service should do is figure out who were the 2 people, study the response, what happened, what was the reaction, and why did those 2 people who had a anaphylactic reaction to vaccines previously, why were they allowed to get this vaccine in the first place is really beyond me. So the problem is the National Health Service. They didn't screen those 2 healthcare workers who had a previous anaphylaxis to previous vaccines and went ahead and gave this vaccine to them without either pre-medicating them or managing their allergic reaction. And then they come out and issue this, huge communique. I mean, this is how the British work. And I think it's absolute nonsense. It's nothing but nonsense. So are we going to be careful about people who have allergic reactions in the past? Of course we are. We do that with any new vaccine, not just with this vaccine. But we now have to do damage control to try and tell people that, listen, don't worry, it's not a big deal. We will take care of you. We will screen you for allergic reactions. We'll manage those depending on what your allergic reaction is. But this is how, politics plays a role in how we roll out an excellent vaccine like this Pfizer vaccine. So you can see the immunogenicity is excellent. And they have the Th1 dominance, which is the good one. And then here is the overview of their phase 2 and 3. I'm just going to show you that they actually selected 40% of their patients were in the 56 to 85 age group. And this was the vaccination period, day 1 and day 21, with a follow-up of 6 months. At least they already have the 3-month data. We already actually saw in the preliminary studies, the 4-month data, it looks pretty good. The neutralizing antibody levels are not declining with the Pfizer vaccine. They are declining somewhat with the Moderna vaccine. I think I'll show you that. I put that in here. And their Th1 cell responses are absolutely spectacular. These are the active ingredients of the Pfizer vaccine, the nucleoside modified messenger RNA encoding viral spike glycoprotein, along with some lipids. So you can have an allergic reaction to any of these. And we know that. That's basic science. Unfortunately, that's basic science for most of us. But for the National Health Service, that's not basic science. To them, this was a surprise. Wonder why. So these are the 2 mRNA vaccines. I just put it together, a single slide. And this is from Pfizer and from Moderna directly from their website. And you can see they look pretty similar, at least at 3 months with the neutralizing antibody response. The Pfizer one is a little higher. But what I think will separate these 2 is going to be the cytotoxic Th1 cell response, which is the lasting immunity for COVID-19. And this is the Moderna one. You can see that the volunteers are 22% are healthcare, 27% have comorbidities, and I showed you 40% are higher than 55 years of age. So it's a well, spread out model of 30,000 patients. With the Pfizer vaccine, it was 43,000 patients. So it's well represented, and it's a cross study of population where this vaccine has been proven to be effective at least at 3 months. These are all the vaccines, human trials going on in the United States. I listed them all here. I'm not going to spend too much time on it. The one that, of course, I'm most excited about right now is Novavax, and then the Merck vaccine, to me, actually may be a slight incremental advantage over all the others. So these are the ones that are also actively recruiting in phase 3. They're not in phase 3 yet. You can see the Novavax has started recruiting already. All the other ones, that I'm talking about right now are probably not going to make it in the United States. They just don't measure up to the new standard, the new standard of care that Pfizer and Moderna and possibly Novavax are going to set. So how do you evaluate vaccine candidates? You talk about durability. We only have 3 months worth of data, in some cases 4 months. We'll see what happens at 6 months. That'll give us a much better snapshot of where these vaccines may separate out, or are both of them, both mRNA vaccines, are they going to perform the same? I don't think so, but that's just speculation on my part at this time. Then storage and distribution, of course, is an issue. In India, how do you store things at -94? I don't know. But in the United States, it's not a big deal. I already got the first set of vaccines delivered at my hospital this morning. We are starting our immunizations this evening. No issues whatsoever. We have a -70°C freezer. The nurse is going in at 4 o'clock today, and she's going to do 4 hours of immunizations for 16 people already. Then Monday, we start again on Monday, then Tuesday, then Wednesday. We have no issues with this storage. This vaccine was shipped in dry ice. It had a chip that monitored the temperature. And I can tell you, as of this morning, I was told that all the temperatures in all those packages that were delivered to our hospital were perfectly fine. So T-cell responses and so just to show you that in a natural infection, I think this is important to understand. In a natural infection, what happens to the spike protein antibody and the nucleocapsid antibody, and what happens to the cytotoxic T-cell responses. It is very interesting to me that when you compare numbers to numbers, vaccine is inducing a much higher neutralizing antibody response and a much more robust cytotoxic T-cell response. At least the Pfizer one is. And I know that the J&J one is as well. So how is it possible that a vaccine is generating a much better, much more robust immune response, both neutralizing antibody and cytotoxic T-cell? To understand that, you have to understand this virus. This is not a naturally occurring virus. This virus not only downregulates the good part of the immune system, it upregulates the bad part of the immune system. And that is why I think this is the most persuasive argument at taking the COVID-19 vaccine. A virus, when it infects you, downregulates your good immune system and upregulates your bad immune system. And that is why, as Satish will tell you, there is an imbalance between the interferons and the interleukins. Alpha interferon goes up, beta interferon goes down. Interleukins go sky high. But those are the bad interleukins. Whereas with the vaccine, what you're seeing is a robust neutralizing antibody response. Why? Because the immune system has not, the good immune system has not been downregulated. So the protection from a vaccine, I believe, a vaccine like the types of Pfizer and possibly J&J, is going to be much longer lasting than the natural infection. And that is why you're seeing reinfections in people who have already had an infection. This is a very, very important concept, which I think all of us need to understand. And perhaps the most persuasive argument for taking the Pfizer mRNA vaccine is because of this. So in summary, duration of T-cell responses, 100% of individuals with prior severe and 87% with mild demonstrate specific memory T-cells. With the vaccine, you're looking at 100%. And that is, I think, because of the effect of the virus on the immune system and its adverse effect on the immune system. The important thing, I didn't talk about this, but here is the reinfection model in a rhesus monkey, where if you do have a robust T-cell response, a cytotoxic Th1 cell response, you are protected against reinfection. So this is, again, animal studies, but a rhesus macaque model in immune system actually does mirror the ones that most humans have for the most part. And so I think what is important here is to understand that this vaccine, the Pfizer vaccine that we have, and a couple of others that are going to follow, which will have both neutralizing antibody and cytotoxic T-cell responses, are actually better for us than getting a natural infection. We have never ever in the history of mankind had a disease burn out by natural infection. Not measles, not smallpox, not any other, not polio. All diseases that we have eliminated for the most part have been because of vaccines. This, I think, is one of the best vaccines we've ever seen for what I think is a man-made virus, a synthetic virus. It's not a naturally occurring virus as far as I'm concerned. And it does, this vaccine, this mRNA platform, has for the first time been validated outside of malignancies where it has been used for the last 4 years. So in the infectious disease world, this is the first time we are seeing an mRNA vaccine. And will this then translate into other infectious diseases? I don't know. Because remember, there is a phenomenon of antibody dependent enhancement of the infection. We saw that with dengue, we saw that with measles, we saw that with Zika, where the mRNA platform did not work. But the mRNA platform for COVID-19 is a perfect fit. So don't translate this mRNA platform as the be all and the end all and the holy grail. It's not. It has failed in other infectious diseases. But I think based on the initial data, the mRNA platform for COVID-19 is going to be a grand slam home run in the bottom of the ninth. That's baseball. But in terms of cricket, it will be a 6 on the last ball of the World Series final, Dhoni style. So that's my analogy of the vaccines. I just want to mention a few things about interleukin 6, anti-interleukin 6 antibody or JAK inhibition, as we talk about. And you can see here this Tocilizumab interleukin 6, which was a total failure in most of the trials because the trials were done again in the wrong way. The dose was given at the wrong time, either too early or too late, and it didn't work. The half-life is very short. So you have to give it at the right time. And when you give it at the right time, for example, in this study in Spain, it actually performed fabulously well. You can see the survival benefit. And then the JAK1, JAK2 inhibition is based in this I think is going to be the future, how to prevent a cytokine storm in someone that has early pneumonitis. If you wait for the cytokine storm to already occur, and the first phase and the second phase cytokine storm, which is the worst of them, of the two, the second one is the worst. If you wait for the cytokine storm to already occur, JAK1, JAK2 inhibition is not going to work. And this is the basis of JAK1, JAK2 inhibition. It's well published, and I talked about this, alluded to this last time. Baricitinib and ruxolitinib, both of them work just as well. And here you are, the proof of concept with rapid reductions in interleukin 1 beta, interleukin 6, and TNF alpha, which is the bad interferon. You can see that this is going to be the way where I think we prevent cytokine storms and improve outcomes down the road. So this is baricitinib, ruxolitinib studies clearly showing that if used at the right time, day 5 to day 6 of infection, day 7 maybe, but before the cytokine storm, the early cytokine storm of the pneumonitis occurs, if you give it before that, you can prevent and ablate the cytokine storm, and the patients will not go down the tubes and not get onto mechanical ventilation. So I'm going to stop here, Satish. I'll hand it back to you. I think I've covered a lot of data in a short period of time, and I've talked for a long time. So you take over, and I'm happy to discuss this as much as you want.
Thank you, Dr. You have wonderfully done your job, and you have given a very comprehensive coverage. And I believe that it was too much in too short of a time. And I hope our audience have been able to assimilate the essence of it. And what I feel, do we discuss the things right now over here, or we let the other speakers speak first?
Dr. SK, I think Dr. Anupam to speak, and we can take up the questions
later on. I request you, all of you, to kindly jot down your questions, and we take them one by one. Over to Dr. Anupam. Dr. Anupam is a very senior physician of our East Delhi, and he has been very active, and he has a special interest in managing the, I would say, rather than the COVID patients. He has been able to manage more than 300 patients over the last few months, and he has been working with the Arogya Hospital in Vaishali, and he has special analyzing the data across the various journals, and he has a very analytical mind. And let's hope what he has to say about the vaccines, because Dr. Moorjani tried to present a view from the American point of view, though it might be applicable to the whole world, but we have at India are likely to have a different scenario. The Pfizer vaccine may not be available, though it may be the best. But what Dr. Anupam has to say about the other vaccines which are likely to be available in India. Over to Dr. Anupam Kumar Singh.
Hello. Am I audible?
Yes, Anupam, you are audible.
Hello.
You are audible, Doctor.
Okay.
You are audible. Kindly unmute your, start your video also.
Okay. Okay, so my internet was to share the screen. Is the screen visible? My screen? Hi.
But visible.
Okay. So I'll be talking about the India vaccine story so far. Okay. So first is the co-vaccine. We have various platforms in India. So firstly, we are talking about co-vaccine, which is an inactivated vaccine. The strain was developed by ICMR, NIV in collaboration with NIV. And these were the trials which were started in India first. So this is an vaccine is an inactivated SARS-CoV-2 vaccine. And the initial plan was to run a phase 1 trial followed by a phase 2. Initial phase 1 was pretty large from phase 1 normal standards. It was 375 healthy volunteers. And then thereafter phase 2 and phase 3. So phase 1 trial was 375. And we had good immunogenicity data on phase 1 trial. So phase 2 trial was shortened from 760 to 380. So according to, Bharat Biotech releases, the seroconversion was as high as 90% in these cases. So they did the phase 2 size. While they have not released the data public domain, but since the data was good, that is why DGCI, they approached DGCI in October, where since they didn't share the data, so they rejected permission for phase 3 because, but then they shared the data again in early November. So on the basis of the seroconversion, up to 85 to 90% seroconversion, the source granted to phase. Recently, they have an early number around 4 December. The UA was refused because they hadn't shared the phase 3 data. So they plan to, conduct, include around 6,000 patients. 2,800 have been recruited so far. So presumably, after the data has been shared, the government will consider an EUA. Talking of inactivated vaccines, it would be useful to talk about other inactivated vaccine candidates. For example, China, Sinopharm. Already started. So, the platforms tend to in details similar, like mRNA platforms are pretty similar in nature.
Your net is not working properly. You can join from your mobile probably.
Okay. Is it working? Hi.
Yes, fine now. Just
okay. So there were some controversies initially. India, a city of brilliant India, country of brilliant individuals, but faulty institutions. When Covaxin first came, an August 15 deadline was given that you have to, it was very confusing. It was the deadline was from ICMR. So the deadline was that the vaccine has to be launched and the trials has to be finished by August 15. I mean, it was very improbable. So there was a lot of controversy. Fortunately, the vaccine, the investigators clarified. Secondly, only animal data are available as preprint, though phase 1 and phase 2 data are out and they have not been shared in public domain. What we have is available is through press releases. So this is a bad, always a bad thing. for example, and that is why, in fact, DCGI didn't allow phase 3 trials in early October. They asked for the whole data, but unfortunately, it is not out in public domain. So that is a bad sign as far as trust in vaccine is very important. So that is why at least you should have some information about when you are asking for an EUA. So at least the phase 1 and phase 2 data from human trials should be in. The animal data preprint was shared in September on a, but yeah, it hasn't been published, but it is available at the preprint. Then third controversy started when a health minister of Haryana tested positive after second dose of vaccination. Then the government said that he was enrolled in placebo arm. So, but it was very confusing as how did they, break the randomization and how did they come to know? So there were some concerns that, so the concern was more about how the randomization was broken and immediately the government came to know. So that also, dilutes in a developing country like ours, which is a democracy and full of conspiracy theory, that actually dilutes, that it is all about trust. So there was a lot of confusion around. The third issue was viral pneumonitis. Viral pneumonitis, post-vaccine, had happened in month of August. It was leaked early into press before the investigators shared the details, to the regulator. So that was very, embarrassing, and so these were, some controversies associated with Covaxin. Some of them, as you can see clearly, are hyped up by the media, but vaccine is a very sensitive issue. The trust should be important, particularly as there are other candidates as well, which may try to hamper your vaccine or play into media, play an opposite controversial media release. Now coming to the AstraZeneca vaccine, which is an adenovirus platform, and the trial is conducted in India by Serum Institute of India. So as we all know that there was a 2 doses controversy with AstraZeneca vaccine, and inadvertent low dose was given in one arm, and it had a higher efficacy of 90% as compared to 60% efficacy in full dose. And a fawning British press actually lapped it up till it was discovered that, okay, there was an inadvertent error. That is why it is not getting an EUA in US, and they plan to conduct a next other trial at low dose. The Indian arm is a full dose. The full dose efficacy as per the UK and Brazil arm is around 60%. So that is what it is. Serum Institute has completed the enrollment of all 1,600 participants on 31st October 2020, and they are awaiting the entire efficacy neutralizing titer data. So again, when they approached the regulator in early December this year in India, they were asked phase 3 data, but they hadn't that with them. So the government has asked them to come again with the phase 3 data as with Covaxin. But the good thing about SII as compared to Covaxin is that the UK data and Brazil arms have been published in Lancet. Obviously, there are controversies as with the randomization. They did a 2:1 randomization and regarding the looks plus. But still, I mean, we have some amount of data. A controversy ensued in India when there was an encephalitis in Chennai, where, while company reported properly, but in response, the company countersued the participant. Again, I mean, it is arguable if they could have, they could have done it more diligently in a proper manner. These controversies, could be, manufactured as well as by various, modified elements, but it doesn't do any favor when there is a, a cascade of suing and counter-suing going on when a vaccine is being rolled out. Pfizer, Moderna, as Dr. Moojani discussed, we don't have any Indian arms. We do have an mRNA candidate here in India by Genova, by a Pune-based company, which has just been granted a phase 1 trial. We know that Pfizer, Moderna have promising international data. There's no Indian arm. These all vaccines, lipid nanoparticles, they have almost 90 to 95% efficacy. Cold chain issues are very prominent. An important issue is these vaccine candidates are not very much interested in India. Is it due to the cold chain? Because while for Pfizer, when all other companies approached the regulator, even Pfizer approached it, but it was wasting time. It said that it wanted more time to make a presentation. When Pfizer has the largest trial which is around, right, and which has the most promising data. It has been granted. UA was granted in NHS first by MHRA, and then in US 2 days back. And Moderna vaccine is due to be, probably next week. It will be granted UA in US as well. So, but are these vaccines, which are standard of care, interested in India? So that is also an important aspect. Obviously, and running a cold chain out of metros is always going to be challenging. Then other vaccine candidate on adenovirus platform. This is Sputnik, the Russian vaccine. So the Russian, they decided in collaboration with Dr. Reddy, they decided to run a trial in India, phase 2+3 adaptive trials. For example, they are simultaneously going on. So phase 2 had 100 patients, phase 3 is planned to have 1,400 patients. While it was planned in October 13, the first doses were received just a few days back in December. The Russian data has been published. It is positive, around 90% efficacy, but very few number of candidates. If I properly remember, it was 30 to 40 infections. So obviously, it is smaller set of data of only phase 2 data. So it didn't inspire confidence. It didn't help that when Russia wanted to roll out the vaccine in its population without phase 3, and the public had very low confidence in it. So they decided to do a phase 3 now. Recently, the AstraZeneca and Sputnik, they are collaborating because they both are based on adenovirus platform. So the idea is to give a Zeneca vaccine first, and then followed by a second dose of Sputnik. And they are trying that out in Russia and some other countries. Now coming to Zydus vaccine. Zydus is a DNA vaccine. So it had completed enrollment and dosing of 1,000 volunteers in phase 2 clinical trials till now. But given other candidates which are very advanced in almost phase 3 stages, probably Zydus is a bit circumspect now. Now it is, as I can see, it hasn't approval. It has sought approval, but it was not very forthcoming with all the even phase 2 data. So they are still awaiting phase 3 approval. They are more interested in this nebulized interferon, the PEGI-HEP trials, which are going in on COVID, and they are pushing more towards that. So Zydus has fallen short as to service its initial promise. Now this is a Genova mRNA, that is the first mRNA candidate from India, that is from Pune, and it has been granted phase 1 trial approval. Just now, probably the trials will start from December. So yeah, so this is the India story roughly.
Thank you, Dr. Anupam. And now we take on to the questions. And before I go on to the questions, I have some challenging issues which are mind-boggling and which are really of great concern to everybody. One is that the mRNA vaccine is being said that it might not hijack the immune system. It might not hijack the genetic code of the individuals. And we do not know that we are able to check the protection of the spike proteins in the individuals who receive it. And there also have been the reports in the past that the mRNA vaccines have been producing eye interferon, which is associated with the autoimmunity also. Whether these vaccines will lead to the autoimmune phenomena in the recipients is another thing to be seen. And second important thing, I'm trying to raise three issues. One is that the mRNA may not hijack the genetic code of the individuals, and it might not turn out to be a disaster like the thalidomide disaster in the past. Second is that the mRNA vaccines in the past have been shown to induce the iron interferon production, which is associated with the autoimmunity. Whether this mRNA vaccine will also behave in the same manner is yet to be seen. Third is the COVID virus in itself causes thromboembolism, and the mRNA vaccines in the past have been associated with the increased thromboembolic phenomena. How do you respond to all these three things, Dr. Moorjani?
Okay, so the question of rogue mRNA, absolutely, that is the biggest concern of all. What if this mRNA goes rogue? How do you then go about stopping it? Or if it produces way too much protein neutralizing antibody and leads to a macroglobulinemia or whatever else that might happen, only long-term studies will show that. And if it does happen, you have to have a way, an antidote, so to say, to turn off this mRNA if it goes rogue. And also then you raise the issue of hyperstimulation of the interferon. So far, what we've seen in the first 3 months of data, that phenomena is not occurring with this vaccine. Doesn't mean it won't occur with other mRNA vaccines, but at least with the Pfizer vaccine and the Moderna vaccine, we have not seen this hyperstimulation of interferon. So the long-term effects need to be observed, need to be watched for. We are doing ongoing studies. This is an emergency authorization. It's not a full approval. So the companies that Moderna and Pfizer cannot do advertising. They cannot push this outside of the strict label that they've been issued, and they have to collect the data. So there's a federal register for the entire population in the US. And it doesn't, there's all kinds of politics about the federal register. Can you put illegal aliens, people in there? Absolutely, you can. So the Immigration Naturalization Service has promised not to use that data to deport illegals from this country. But that's politics. Besides that, the two companies will get long-term side effect, adverse event, and efficacy data. So we'll see what happens at 6 months, 9 months, and 12 months. But these are valid concerns, Satish, where you're talking about a rogue mRNA and what that can do to you. And there was a question about what about vaccines and people who have already had the disease. I already alluded to that. And I think it makes absolute sense that you take the vaccine, because if the disease has been mild and your immunity is not as robust, you will get a reinfection. And most of the reinfections that we have seen so far, if not all of them, have been mild, proven reinfections. But you don't know. And the vaccine, as I said to you, is not the whole virus. So it can only potentiate the robust T cell responses and the neutralizing antibody response you've had. So I would encourage those. And for example, in the Moderna and the Pfizer trials, 11% and 13% respectively of all the patients that were included, retrospectively, when they were looked at for antibodies, they already had a subclinical or a low clinical evidence of infection in the past. So those people also got the vaccine and they are doing fine so far. That data was just released yesterday, where people with pre-existing positive antibody, when they got the vaccine, what happened to them? They've all done just fine. There's been no adverse outcome in those people as well. So yes, take the vaccine if you've already had the disease, especially if you've had mild disease. What about severe disease? Be careful. What was the third, Satish, the third part of that question?
The possibility of getting increased thromboembolism in these patients because of the mRNA.
Yeah, so clearly, we know that the way this virus works is that it resets your inflammatory pathway and it resets your thromboembolic pathway. The pathways have now been elucidated nicely. I think in the slide set I sent to you, Satish, I've hidden those slides about thromboembolism and the pathway of thromboembolism. But the vaccine is not expected to do anything of that sort because it doesn't affect that pathway, period. All it does is create neutralizing antibody to the RBD and to the fusion antigen. So prevents the formation of the fusion channel, and it also creates a cytotoxic T cell response, but that cytotoxic T cell response is very different than the thromboembolism. So thromboembolism is not an issue with the vaccine. What is important from the thromboembolic perspective is that if this mRNA goes bad, let's say it's sitting out in the open at 30 degrees centigrade for 1 hour and then somebody injects it into you, that mRNA can lead to localized thromboembolism at the site of the. So I've said this, that degradation of the mRNA can occur as soon as that temperature starts falling below 70 degrees centigrade.
Harish,
So,
I'll interrupt Dr. Pankaj. You kindly mute everybody. There's a lot of noise.
Dr. Gupta, you are the host actually. No? You can do it at your end.
Hey, Pankaj, I thought you were not going to be here today.
Actually, I was planned to travel, but I could reach in time actually.
I don't know how to host. How do I stop everybody? Like
Dr. Piyush Chadda, your mic is cause disturbance. So kindly mute yourself. Dr. Piyush Chadda.
Hello.
Kindly mute yourself, Dr. Piyush. Today, Skipta, you can go to the participant who is unmute. You can long press and you can mute. Rajesh, carry on.
So, when the mRNA gets denatured and you inject that can lead to thromboembolism, at least locally. So I tell people that, if it's a theoretical concern for you, then take aspirin and turmeric and saffron 5 days before the vaccine and try and downregulate your immune system that way. Again, there are studies on aspirin and its effect on efficacy of vaccinations in the past, but with mRNA vaccines, especially with this neutralizing antibody vaccines with the Pfizer and Moderna one, there are studies that have been done with aspirin and these vaccines, and it's been shown to have any impact whatsoever. So there was a nice study published in the journal Science on this issue. And so I'm very happy to take aspirin. So I've started already taking 162 milligrams of aspirin, and I usually take my turmeric and saffron anyway. So I hope I'll let you know in a week or so.
But Harish, have you taken the vaccine shot?
So I've scheduled myself for Wednesday. I could have taken it today, but the next 2 days are very busy for me, Monday and Tuesday in my office. And I didn't want anything to create a obstacle there. Plus, remember, I have 4 infectious disease people in my group. So one is taking it today, one is taking it Monday, one is taking it Tuesday, and I'm going to take it Wednesday. Because we don't want people to, all 4 of us to take the vaccine on the same day, and then all 4 of us can't function. That would be a disaster in this time. There's more than 150 people admitted to the hospitals we go to with COVID right now. So we need all hands on deck. So for those logistical reasons, I will take it on Wednesday. Wednesday morning is my scheduled date. And then I'll report to you. But valid concerns, Satish, you're talking about...
Which vaccine we're taking? Pfizer?
I'm taking the Pfizer. That's the only one available in the United States right now. Moderna will be available in 2 weeks. So we'll see who prefers what.
So there are 2 questions in the chat box. One is the issue of vaccine in neurological disorders like multiple sclerosis. Any issue?
Yeah, so there are no studies.
Pankaj, I'm going to put this question in the way. There are 4 things which have to be seen in any vaccine before we embark upon taking this vaccine. One is the issue of transverse myelitis. We have already seen 2 cases of transverse myelitis with the Astra vaccines. We have already seen 2 deaths. You might have tried to just pass on the buck with saying that they did not choose the participants properly. But when you're going to vaccinate the huge population, and it will be really very difficult, particularly those who are less literate or those who are not aware about the allergy, and you still like to go ahead with the risk. And third, the autism and the multiple sclerosis and later on the Guillain-Barré syndrome. And how do you go about analyzing this risk with these various vaccines, particularly the Astra vaccine and the Pfizer vaccine?
Okay, so the issue of 2 deaths. So there were 2 deaths in the Pfizer vaccine arm. Compared to placebo, in the placebo arm, there were 4 deaths. So the one way to analyze this is the excess mortality. So when you look at 43,000 patients or 30,000 patients in the Moderna vaccine arm, what is the expected death? Because remember, these are people who are 80s, 85, they have comorbidities. Some of them will die from things. I mean, that's natural, that's life. So what is the excess death? So there were 2 deaths in the Pfizer vaccine arm and 4 deaths in the placebo arm. Were the 2 deaths related? The answer is no. Both of the deaths were natural causes, not related to the vaccine or vaccine adverse events to begin with. And the excess death number has not been calculated based on that life table. But I think once we have 6 months of data, we will know the excess death rate because it takes about 6 months for the life tables to be sort of correlated. So that's one part. The other part is, what about people with Guillain-Barré syndrome and neurological disorders? There you have to be careful of any vaccine period. And all those people were actually excluded from these trials. So I would advise everybody with underlying neurological disorders such as multiple sclerosis and previous history of Guillain-Barré not to take this vaccine or any vaccine for that matter, unless they talk to their provider and they do the risk benefit analysis for themselves and figure out for themselves whether they want to take the risk or not. There is a clear risk of Guillain-Barré syndrome and multiple sclerosis getting worse with any vaccine, not just the mRNA vaccine. And we have no idea what happens to people with neurological disorders when they get an mRNA vaccine. It could end up that the mRNA vaccines are contraindicated. They have not been studied. I would tell people not to even bother. What about the adenovirus vaccines? We know they're associated with transverse myelitis. We know they're associated with encephalitis. The issue being, is it 1 in 1,000? Is it 1 in 5,000? Is it 1 in 10,000? 1 in 100,000? Only large-scale trials with the COVID vaccine, adenovirus vector vaccines can answer that question. But I would be very, very careful. If we didn't have a choice, I think adenovirus vaccines make sense. But now that we have a choice now, and we have potential options in future with adjuvant vaccines that are much better than the adjuvant vaccines we have now, I think this issue will become moot. And I think if you're going to do an adenovirus vaccine, you're going to then have to look at which adenovirus vector is the safest one. Is it going to be the chimpanzee adenovirus? Is it going to be adenovirus 5? Is it going to be adenovirus 6? Or is it going to be the J&J adenovirus combination that they are using with one shot? Is it going to be 2 vaccinations? Is it going to be 1? We know that the Sputnik 5 is 2 vaccinations. Now they're combining with AstraZeneca because AstraZeneca has a chimpanzee adenovirus and Sputnik has adenovirus 5 and 26. They want to see what happens with chimpanzee and adeno 5 in one combination and chimpanzee and adeno 26 in the second combination. Do you get better responses? We know adeno 5 antibodies can mute your immune response. So is it better to go with chimpanzee and adeno 26? Only time will tell. But the bottom line is all this is going through a very huge amount of study right now. Answers will come. In the meantime, if you're worried about transverse myelitis and you talk to your provider and you come up with this whole risk assessment where your risk is small, but your benefit is huge, then maybe you want to go ahead with the vaccine that's available in your part of the world. If you're in the US, I would have no problem in someone to give them the mRNA vaccine, but not the adenovirus vaccine if they have neurological issues in the past. But again, that's my personal advice to them. It's not, and if the patient says, no, I'm not ready to move on with it, I don't want to take any risk, then fine, just keep doing what you're doing to protect yourself from COVID-19. So it's always individual risk assessment, risk and benefit analysis that goes, that has to take place. What about autoimmune disorders? That's a clear risk. Again, autoimmune disorders, people with underlying connective tissue disorders were excluded from these trials. And I would not want them to take any vaccine unless there is data. And the same analogy applies there, where you have to sit down with your doctor, your provider, and then do the risk-benefit analysis and see if you want to proceed. What about pregnancy? So we are starting trials in pregnancy in January with the Pfizer vaccine. So right now, there's no data. We are also starting trials in people in age group of 12 to 16. Remember this, the Pfizer and Moderna trials are all with 16 plus. There was a question raised about the fact that 16 to 18, there were small numbers. 16 to 18 age group is the same as far as I'm concerned. I don't see any reason why there should be any difference in that situation. What about renal transplant? The mRNA vaccine in transplant patients is no issue whatsoever. Renal transplant, liver transplant is not expected to have any side effect. Maybe the benefit will be less because these guys are immunosuppressed, but then even more important that they get the vaccine so they are protected because they're sitting ducks against this virus. This virus comes into a immunosuppressed or a transplant patient, and we've seen that. As recently as over the last 3 months, we've seen several transplant patients present to us. And over the last 3 weeks, we've been giving them the monoclonal antibody, and they've all done well. But before that, it was a disaster. Transplant patients with COVID-19 was a nightmare. And I would highly encourage those people to go with the mRNA vaccine. Should they go with the adenovirus vaccine? Probably not. because there, even though it's a adenovirus vector, which is attenuated, it could lead to problems there. So again, it's an individual assessment, Satish. But I'm telling you, I think you're right on. mRNA vaccines are brand new, and not much is known in long-term effects of these vaccines. And one has to be very, very, very careful about taking this at an individual level. I'm going to take it because my risk is so high. And I am hoping that Pfizer, and I actually know that Pfizer has the antisense technology to turn this off if and when the need arises. They are ready for it. Everybody's talking about it. They are also talking about it. They've researched it. They're actually doing studies on this. How do you turn this mRNA off? And I think I know people who are working in Pfizer who express confidence that they are very close, if not already there, to turning off the mRNA if they have to. So I hope that answers those questions.
Somebody has asked that certain people with certain diseases who are on immunosuppression should they go with the vaccine?
With the JAK1, JAK2 inhibition, again, not much known in immunosuppressed, but yes, my bias would be that if you are presenting with COVID-19 and you are in early pneumonitis phase, day 7, and the interleukins are normal and C-reactive protein is normal, I would treat them with baricitinib or with ruxolitinib. Yes.
Fine, Harish, we wish you all the best with your vaccine, which you are planning to take in a day or so. But I have another question. See, Pfizer vaccine, though it may be an excellent vaccine, but it has two issues with it. One is the cold chain, and what I understand is that if the vaccine comes to India, we have an immense capacity to manage the cold chain, and with our Prime Minister being so dynamic and active, we'll be able to handle this issue. But the another issue is the capability to manufacture. The Pfizer has a limited capability to manufacture, though the vaccine is likely to come to India, but it is going to be available only to the capitalist people, the corporate managers, and the celebrities. Though it is not likely to come to me and Pankaj or some of the people around. We are likely to get something like Astra vaccine or something like Sputnik vaccine. And if we wait about 6 months or so, we are likely to get Novavax vaccine. I say Novavax vaccine because the Novavax has a big agreement with the Serum Institute of India to manufacture nearly 50 crores doses, but it is going to be around 6 months from now. So what do you advise, that we should wait taking the vaccine, Astra vaccine, or the Sputnik vaccine, or we should go ahead with taking these vaccines, which I believe they are not very effective, and there have been issues, and there have been some fudging the data or some suppression of the data by the Astra particularly, and the Sputnik data is still not in the public domain. It's not very convincing. And the Novavax vaccine, about 6 months from now, the doctors, the forum doctors are already apprehensive. We had done a survey wherein about 45% of the people said that they are going to wait. What do you say, that we should wait, or we should, in case we feel that we are into the high risk order, we should go ahead with the Astra vaccine or the Sputnik vaccine, and then go with the second dose of the Novavax vaccine?
Yeah, so I think these are all, again, excellent questions. Can you go from one type of vaccine to another vaccine in 6 months, and will that have an adverse event? Again, no answers, but I can tell you based on the knowledge that I have, and my speculation here is that you can go from an adenovirus vaccine to an mRNA vaccine. You can go from an adenovirus vaccine. You can clearly go from an adenovirus vaccine to an adjuvant vaccine. There's no question in my mind, because the potential for any intervention by an adenovirus vaccine against a subsequent adjuvant vaccine is practically zero. And we've done that before in other diseases in the past. And these are proven technology. This adjuvant vaccine from Novavax, the M1 protein adjuvant, is just so much better than the aluminum adjuvants and all the diphtheria adjuvants that we used to use in the past. So clearly, we can go from adenovirus to adjuvant. Can we go from adenovirus to mRNA in 6 months from now? Don't know. And the reason I don't know, and I say I don't know, is because of this whole issue I alluded to, antibody-dependent enhancement. Will that happen with COVID-19? So far, we have not seen it. If it doesn't happen, I think you're perfectly fine. But that's the big concern. We saw that with dengue, we saw that with Zika, we saw that with measles with the mRNA platform. So that's the only concern I have. You cannot go from adenovirus to mRNA unless it's proven in COVID-19 that you don't have antibody dependent enhancement. Okay, so I think that's just speculation on my part. All these questions will be answered in the next 6 months. So what should an individual like Satish do, or Pankaj do, or I should do if I was in India, or so many of our friends from our group, from 81 batch, or for that matter from our entire college, what should they do? The risk is very high for you guys. The risk for me is sky high. I mean, look at me. I've just recently crossed 2,000 patients with this second surge that I'm seeing since November. I mean, every day I see 30, 40 patients with COVID. It's sky high. The risk is absolutely sky high. If I was you, I would take the Sputnik 5. I would actually try and get the J&J vaccine. That should be released very soon. I don't know what the status in India is, but the Sputnik 5 vaccine to me looks fairly good. Now it's a Russian vaccine. The data is sort of hedged and it's not transparent. There's no transparency in that data, but the science is valid. The science has been proven. It's not as good as the mRNA, but 80%, 75% response is better than zero. And you have to do that for yourself individually. You know, are you at risk, and what will happen if you are at risk? Will you be able to catch this early and treat yourself early? You know, I can't advise you personally, Satish, Pankaj, but I think the default should be that you take the vaccine, whichever one is best. I would not touch the AstraZeneca vaccine. That I can tell you. They've fudged the data. They've made a mess out of it. Stay away from that. The Chinese vaccines, you know, I'm always concerned about anything and everything from China, including stuff that comes out of there by way of science. I would not. That's my personal opinion. I would not touch anything that comes out of China. And you know, I think you're fine with the Sputnik 5. That's my best advice right now at this point.
That's great. And I would like to
I do want to say something. Yes, I forgot. So Pfizer has limited, at least they've publicly admitted that they have limited production capabilities. I can tell you that they are going to ramp it up. And ramping up these production facilities takes maybe a month or two. And they've already seen over the last 1 month the interest in the Pfizer, in their vaccine platform. And I think if I know Pfizer and their management well, they're probably already gearing up. By March or so, they should have ramped it up even more. I know, for example, Novavax has already got a collaboration with the Serum Institute. They are launching phase 3 trials. I think by March, you should have the phase 3 data with Novavax. With 3 months, and I think you can go with that. And that's the one that I'm most excited about. So if you can wait 3 months and protect yourself like you have done for 1 year, then do so. If not, take the Sputnik 5 and then take the Novavax.
That's great. I personally believe that I would love to wait for the Novavax because the antibody titer has been nearly twice that of Pfizer vaccine and about 6 times that of Moderna vaccine. Right. So it is going to be interesting to see their phase 3 data. Till then, like whatever is available, one has to make a choice. We do not really know whether will be the frontline workers, so-called VIPs right now, but in fact, more of guinea pigs backstage. We have to be really careful choosing vaccine. Yeah. And as of now, I really feel that it has been a great show, and we have been able to answer large number of questions. In case somebody has a question, one may come forward, unmute oneself and go ahead with the question.
I would like to add something.
Yes.
So one Pfizer vaccine group had 4 cases of Bell's palsy. I mean, it could be unrelated to it, but FDA document mentioned it. So they said that the baseline rate of Bell's palsy was higher in this is more than the number of 4 cases in the population. But it was interesting that the cases were seen in the vaccine arm. So I mean, just like a first rotavirus vaccine, you had an intussusception, which was found pretty later. So I mean, that is one neurological aspect of the mRNA vaccine I would be careful about. The second part is that regarding the patients who have already been infected, Pfizer had 1,000 patients who had already had antibodies, and they Pfizer trial, and they actually were enrolled in the trial. And even out of these 1,020 people had reinfections. That is almost 17 to 18 in the placebo arm, and 3, I think, in the vaccine arm. Out of which 2 were, before the second dose was given. So the point is testing for antibody before taking a vaccine a good strategy? I mean, personally, I think it is doubtful because, one, you don't know if your test is false positive because, most antibody tests are, almost 99% specific, but there's the 1% chance that you might be false positive. And as data from Pfizer, subgroup shows that 2%, if it is a true positive, you still up to 2% had reinfections, particularly if they're mild. So, an antibody strategy is perhaps not doing an antibody before taking a vaccine is personally not a very, sensible strategy in my opinion. I personally had COVID disease. If I had availability, I would still take Pfizer vaccine. In India, availability is an issue, but I hope things will get better.
So I think those are all fabulous points. The issue of Bell's palsy, 4 Bell's palsies in the Pfizer arm, the vaccine arm, I think Bell's palsy is so common, at least in the northeast part of United States, where tick-borne diseases are probably the cause of Bell's palsy. And then you have the idiopathic Bell's palsy. And the good news is all those 4 people with Bell's palsy, they recovered. So that's one issue. The other issue I agree with you, from a public health perspective, there's no point doing antibody seroprevalence to decide who takes the vaccine or who not. But at an individual level, and I think especially in someone who has underlying neurological diseases, as Satish talked about, I think there you do your antibody level. And if you know that you're antibody positive and your antibody titers, but again, the problem is we don't do neutralizing antibodies. When we do the test for antibody, we just look for IgG. We don't look for neutralizing antibodies. So to the receptor binding domain. So if you're going to do that antibody test and you rest assured that you've had the infection and you're protected for at least 6 months, even if you get a reinfection, as you pointed out, reinfections are happening now, it's going to be mild and you're going to be fine as long as you take early treatment. Then I think you can afford to wait for the Novavax vaccine if you're concerned about the mRNA or if you're concerned about the adenovirus vaccine. Then I think doing an antibody at an individual level to come to that decision whether you should take the vaccine now or not makes sense, but not at a public health level. I agree. I couldn't agree more.
Fully. I fully agree that it could be a disaster in case the whole public is subjected to the antibody levels and it creates a lot of chaos. Any other questions, please?
I think most of the topics have been covered. Dr. Harish and Dr. Anupam have given a very excellent overview. And as Dr. Harish has rightly said, that we are actually in infancy because we don't know the virus, where it comes from. It's a natural source or man-made virus. But definitely it is altogether different. And how vaccine works, it would be a great debate again. So Dr. Harish has already said that we have to wait for 3 months after vaccine at how antibodies respond. And I think it would depend upon individual levels also. Like some may respond to respond. Some may not respond even. They may not produce any antibodies. As the virus has shown, in many people with COVID, they don't produce antibodies.
That's
right.
Correct.
So
14% of mild infections. Yes.
Yeah, they haven't antibodies yet. Right? So same about the vaccine. I think no vaccine had had potential definitely because it has the highest number of antibodies have been produced by this Novavax. And we have given, Dr. SK has said that we have made a trial in this thing, and 45% of EDP members feel that they should wait and they should get a better vaccine. Dr. Anupam, what about this COVAX? What is the future of Covaxin as of now in India? Do we have some insight into it?
The Covaxin, it's an inactivated vaccine. Only the Sinopharm from China. I mean, we generally see that the similar vaccines from similar platform have around similar efficacy. has 85% efficacy. It is a very limited data is out in the public domain. The only data which I can see is that around 80%, in around 85% of individuals, there was zero conversion, no data on efficacy. The amount of people which developed severe infection. One issue with all vaccine trials is that people modify their behavior once they are in the trial. So you would see a lesser number of severe infections as compared to the other in the normal population. So there is a trial behavior modification, so that you couldn't directly generalize it to a normal population about the number. It is impacting the number of severe infections or not. Till now, we don't have the data from India. I would personally take an mRNA or adenovirus before over an inactivated vaccine. That is my
Right.
Yeah, I couldn't agree more. Volunteer bias, Pankaj and Satish, is a very real thing. And don't try to then compare what happens in real life to what happens in clinical trials. Because again, as Anupam mentioned, people who take the vaccine are on their best behavior and take the best possible measures to protect themselves. So the rate of infections is much less. So we'll see what the real-time data shows once the vaccine
Yeah,
is.
Pankaj, can I ask one question here?
Dr. Paras, go.
Yeah. Sorry, I have roped in just a little late. It was fantastic session today. I just want to ask if the vaccine is available to all, should the youth, say age 30, 35, wait for 3 to 6 months to get the vaccine? Because the disease in them is likely to be mild. I mean, who, if the vaccine is available to all, should be refrained from getting the vaccine now? Should the youth be given the vaccine now?
I think Dr. Paras is a very relevant question, because we see a mild to moderate cases in youth, but definitely there are mortalities in younger population also. So I don't think so that we should restrict them to take vaccines as of now.
I have a different answer to this question. The government of India has already decided that it is the frontline workers who are going to take the vaccine, which amount to nearly 1 crore people. And then it is going to be the social workers, the police, and all the other people, which amount to nearly 2 crore people. And then it is going to be the people with the comorbidities, which are likely to be around 27 crore people. Then come the young people without comorbidities. So your question will be answered by the scenario in the vaccination program itself.
So my question pertains to a different aspect of it. Should the youth be made to wait for 3 to 6 months for good data to emerge before they are given the vaccine? Because in them, the possibility of having something bad with COVID is lesser as compared to something wrong with the vaccination, which is not yet proven. So that's my full question.
But Harish can answer this question.
I'm not sure how to answer that question in a public health setting. So my answer to any public health question and public health initiative is that you've got to look at the local factors. And when you have limited availability and you have to prioritize, then you do so. And you do so based on where the disease is most severe, where is it impacting the healthcare system, and where is the economic benefit of that intervention. Those are the three factors in that order of priority, where I then put my 2 cents in a public health unit. So I've been in public health for 25 years now, 26 years. And those are the ways that we then collectively decide. So there are other people who have different priorities and different aspects. And then you get 10 people or 12 people around a table that understand that and come out with an initiative that you think is appropriate for that local situation. This is called population health. Not
so last lingering on my question, Dr. Moorjani has been kind to send me 5 vaccine shots for my family.
Yeah.
Right?
Yeah.
Do I give including my kids or not now? Or does Dr. Moorjani says give it to the elders, hold on the kids, wait for 3 to 6 months data to come, and then offer to your kids?
Yeah, I think the second scenario is what I would personally do, but that's your personal choice.
Right, that's fine. That's what I agree with.
Yeah, you have to reduce the spread of this virus. Where is the maximum impact of a limited resource? That's the question you're asking, Paras. And I think you're absolutely right. You've got to give it to where it's most effective.
There's second component to that also, that unnecessarily not burdening the kids who are likely to recover themselves with the possibility of vaccine-related misadventure.
Correct. Right on both fronts. I agree completely.
Right. Thank you so much.
Dr. Moorjani, there have been some reports that the vaccine in the Middle East is being given primarily to the migrant population, and the native population has been like spared. What is the scenario in the US and in the UK?
I can tell you my personal experience. I've been given allocation of vaccine to give out, and I'm not discriminating. It's based on risk assessment, and we are giving it to even people who are illegal, who are immunosuppressed. I have patients in my practice who are HIV positive, who are immunosuppressed with CD4s of 100, 200, and they are priority, and a couple of them are illegal. They have a work permit, but they're illegal. They are not citizens. They're not green card holders. And they've been included in that list, and they have made it to that list. So they'll be getting their shots this week.
Do you have any knowledge about UK? Are they prioritizing the migrant population, non-native people?
I do not know. So we've been specifically told that migration status, illegal, legal status, green card status has no role to play in selection of priorities. So, which is a good thing.
That's great.
Moorjani, what Trump is getting the vaccine? Is it Moderna or? Because he already received antibodies.
I don't know. I can tell you Anthony Fauci is going to take the Moderna vaccine because he was behind the whole development of the Moderna vaccine. So he has to stand behind it. And Joe Biden and Barack Obama are going to take the Pfizer vaccine. They've been offered that already. They do qualify. The rest will be all divided between Moderna and Pfizer. My personal bias is Pfizer for the reasons I mentioned. But I think both of them are going to be pretty good. And the question is, I think the big question, Pankaj, Satish, and everybody else is that, can you take the Moderna vaccine and then take the Pfizer vaccine later on? As I alluded to it, I don't think so. I think that's the biggest reason not to take the Moderna vaccine. But that's my personal opinion. It's not an official statement. Okay.
The final question, Dr. Harish, will this virus die off in time, or you have the second shot of vaccine next year?
Great question. Can we kill off this epidemic? I think again, you have to go back to where did this virus come from? How did it originate? And that's why I started the discussion with that. I don't think this is a naturally occurring virus. So and if you believe that, and if you're with me on that, then I think it's almost certainty that we will eliminate this pandemic very soon, maybe by the end of 2021. We have the tools, we have the science, we have the knowledge. There are very, very smart people who understand this, but you have to accept that this is not a naturally occurring virus. And that's where all the knowledge comes from. And that's where all the knowledge flows from.
So hopefully we'll visit US in early 2022.
Oh, absolutely. Yeah. Come hang out with me. I can show
sure.
you.
A debate in the US is currently like how to prioritize the number of doses, because Pfizer vaccine shows that even after first dose you have around 85% efficacy. So do you reserve 2 doses for an individual, or you give one dose to the Scott Gottlieb is already buying Pfizer.
Anupam, that's I think this argument was raised in the Wall Street Journal a couple of days ago. And I have only one word for it: absolutely baseless nonsense. Just total garbage argument. Go with science, go with knowledge. Don't go with this, half-ass stuff, 80% protection, 20% protection, whatever. No way, no chance. No chance in my world. I don't know about anybody else's world, but I consider this a garbage argument. And I said that to Wall Street Journal as well. You will see this on Channel 7 News on Monday, tomorrow. At 5:30 p.m. with Anthony Fauci, where I publicly I'm going to go on record now on national TV saying this is garbage.
That'd be great hearing you for there.
I hope.
And we are all waiting to you to challenge Anthony Fauci.
No, I'm not challenging. In fact, he has the same. I trained with him. I did part of my training with Anthony Fauci. So. He's a man I respect tremendously. So he's going on saying there's no role for this strategy of giving everybody one Pfizer vaccine, because all you're going to do is create a complete chaos and mess down the road. And we are not going to do that. It has to be based in science. It has to be based in knowledge. It has to be structured. And we've taken the risk so far, and we're going to continue to take that risk and do whatever we need to do.
Harish, just a request that if you can make a sort of gist of the representation in a text, so that we can post it in our group, so that everybody would be able to.
Oh my God! I can send the whole PowerPoint to you, and you can export it to a Word format.
That can be done. Yeah, that can be done.
Pankaj, I understand. I understand Dr. Moorjani is very busy, and some of us is going to do that. And I think we should excuse him from all this.
Yeah, sure.
We should be thankful to him that he has been, despite his busyness, he has been kind enough to come to us and give the wonderful data and all the information in black and white.
So Satish has the presentation. He
Right.
can export it to Word.
Yeah, we can present text and then we can present.
I'm happy to do that.
I still don't agree to that, but he had tried to pass the buck.
All right, no more questions, huh?
Right.
I think I would like to correct Dr. S.K. Gupta. It was the most colorful presentation. There was nothing black and white.
So
Well,
I think
it's interesting the colors of my slides matched his dress.
Yeah.
I think this is the time to wind off and say thanks to all the participants and all the both the speakers. Dr. Pankaj, I request you to please give a vote of thanks.
Ashok is here. Dr. Ashok, you can give a vote of thanks. Ashok.
Thank you, sir. It is very kind listening you, and I thank especially Dr. Harish Moorjani. It always been good listening from you and getting knowledge about all the vaccine, but still we are India still get to a proper vaccine, and you have given a very wonderful overview of all the vaccine and various trial and all, and you have given your preference regarding which vaccine you are going to get it by the next coming Wednesday. And I thank Dr. Anupam Singh. He is my colleague who has presented Indian data in the context of vaccine very well. Thank you, Dr. Anupam, and I thank Dr. S.K. Gupta, who has very well moderating the session and conducted interactive session among us. Thank you all, and good night to all of you. Thank you very much.
Thanks so much.
Bye.
So have a nice day, Dr. Moorjani.
Thank you, Harish. Thank you so much.
Thanks, Satish. Anytime.
And we'll wait for you to be there with us again.
And good luck for your shot.